Welcome everybody to today's instalment of our Winter-Spring webinar series. Today we have the lovely Dr. Samar Issa speaking to us on new emerging targeted therapies in lymphoma, which should be an exciting talk. And I'll introduce Dr. Issa in a moment, but for those of you joining us for the first time, we will be recording the session and it will be available on our YouTube channel and Facebook pages shortly afterwards. And so you can go back and have a look at the slides and everything on those recordings. We also have a Q&A box that should be visible at the bottom of your screen. So if you have any questions during this session, please do pop your questions in there. And Dr. Issa will be answering questions at the end if we've got time, which would be good. All right. So as I said, we've got Dr. Samar Issa here speaking for us. She is a consultant haematologist and the clinical head of the lymphoma service at Middlemore Hospital in Auckland, New Zealand. Dr. Issa graduated from the School of Medicine at Baghdad University and then trained in Auckland, where she gained the fellowships of the Royal Australasian College of Physicians and the Royal College of Pathologists of Australasia in 2007. She undertook a fellowship in translational research at the University of California, San Francisco, and her research focused on the central nervous system lymphomas. Dr. Issa is an active member of a number of scientific groups. She is the founding chair of the Lymphoma Network of New Zealand and the founding clinical director of the Middlemore Tissue Bank. She has multiple research publications and is the primary investigator on several local and international lymphoma trials. She has also authored a few book chapters in the field of lymphoma, so she is a very, very knowledgeable speaker on this topic. So thank you very much for your time, Dr. Issa. We're looking forward to your talk.
No problem. My pleasure, Melody, and thank you so much for inviting me to present this webinar. I had talked a few weeks ago on the biology of lymphoma. And that was the first talk in the series of the webinars on lymphoma. Since then, we talked about Hodgkin lymphoma, aggressive lymphomas, and T-cell lymphomas. Today, we're talking about targeting therapies in lymphoma, which is an emerging field and an extremely important topic. So I would like to start by reminding all of us about the basic and fundamental points in what lymphoma is and how chemotherapy works in lymphoma. So to start with, I'd like to talk about what is cancer and why it happens. That will help us to understand how do we target these cancer cells in the targeted therapies that I will describe later in this talk. So cancer cells behave differently than normal cells in the body. Many of these differences are related to cell division behavior. Cancer cells basically divide uncontrollably, and cancer is a disease of uncontrolled cell division. Its development and progression are usually linked to changes in the activities of cell cycle regulators, what we call mutations. The genetic changes or mutations that contribute to cancer tend to affect three main types of genes: protooncogenes, tumor suppressor genes, and DNA repair genes. These changes are sometimes called the drivers for cancer. So what are protooncogenes? Protooncogenes are normal genes. They are involved in our normal cell growth and division. However, when these genes are altered in certain ways or are more active than normal, that means they become cancer-causing genes or oncogenes, allowing the cell to grow and survive without any control. On the other hand, tumor suppressor genes are involved in controlling the cell growth and division. So they inhibit the cell division. And the cells that have certain alteration in the tumor suppressor genes, they start dividing in uncontrollable manner. DNA repair genes are involved in fixing the damaged DNA in normal cells. It happens all the time in our normal cells that the DNA becomes abnormal through the division process and the growth process. So these genes repair our damaged DNA. Mutations or abnormalities within these genes tend to happen sometimes, and that may lead to the development of cancer because of the accumulation of these abnormal or unrepaired genes. To understand targeted therapies, we need to know a little bit about the basics of the tumor microenvironment. Cancer cells can influence the surrounding normal cells, including the blood vessels and other normal tissue cells that exist in our normal tissues, like fibroblasts, T cells, dendritic cells, etc. What happens during cancer development is that the cancer cells start ignoring the signals that come from the microenvironment that tell them to die, which is called the programmed cell death or apoptosis. So they survive and the body is unable to get rid of them. Furthermore, the cancer cells can make blood vessels to supply the tumor with oxygen and nutrient that they need to grow. And then, of course, these blood vessels will remove the waste products from the tumor so the cancer can survive and grows further. What is the role of the immune system in cancer? Our immune system normally is very effective in getting rid of abnormal particles, including viruses, bacteria, foreign bodies, and cancer cells. I talked about that in the biology of lymphoma webinar in detail a few weeks ago. The immune system is made of networks of organs, tissues, and specialized immune cells. These are the T lymphocytes in our body. They remove damaged or cancer cells from our body. They are the police men and women of our body. Some cancer cells are able to hide or evade from the immune system. Furthermore, they can make these T cells work to their advantage, and they can make them protect the cancer cells themselves through secreting certain particles and molecules on the surface, which I will describe very soon. A reminder of what lymphoma is, which is the topic of our talk today. Of course, lymphoma is cancer of the lymphatic tissue. It includes B cells and T cells. These are the disease-fighting white blood cells and are part of our immune system. There are two main types of lymphomas, Hodgkin and non-Hodgkin lymphoma. Hodgkin lymphoma has abnormal lymphoid cells that are called Reed-Sternberg cells. non-Hodgkin lymphoma cells can be B or T lymphoid cells and can be aggressive or indolent, which means slow-growing cancer. There are different types of conventional treatments for lymphoma patients. Sometimes we take the approach of watchful waiting in indolent or low-grade, slow-growing non-Hodgkin lymphomas. Other approaches include surgery, sometimes a limited stage, what we call extranodal lymphomas. If we remove the lymphatic gland that's affected by cancer, we might be able to cure the patients. Or in skin lymphomas. Antibiotics can be used in treating gastric, stomach, low-grade mold lymphoma. Radiotherapy in early stage lymphoma, that is in low-grade lymphomas. Chemotherapy or combination of chemotherapy agents. And then targeted and immune therapy, which I will talk in detail very soon. How does chemotherapy work? The chemotherapy usually targets the dividing cells. And since cancer cells divide very rapidly, more so than our normal body cells, the chemotherapy can destroy the cancer cells. They damage the genes in the nucleus and make the cancer cell unable to divide. And so the cancer stops growing and dies. This is the cell division cycle, and the cancer cells mostly work on this phase of the cell division cycle, which we call the interphase. That's when the cancer cells start preparing itself to divide. During that period, the chromosomes separate and the cell is vulnerable. Excuse me. These are some of the chemotherapy agents that we use in the treatment of cancer. Some of you might have heard of them. They work on the different cell division cycle, and they stop the cell from dividing and causing death. The problem with chemotherapy agents is they target any dividing cell, unlike targeted therapy, which targets the cancer cell only. And that's where the differences between targeted therapy and conventional chemotherapy are. The targeted therapies act on specific molecular targets on cancer cells, whereas most of the standard chemotherapy, as we've just shown, act on all rapidly dividing normal and cancerous cells in our body. The targeted therapies are deliberately designed to interact with their target, whereas many standard chemotherapy agents are identified because they simply just kill cells. They are often cytostatic, which means they block the tumor or cancer cell division, whereas standard chemotherapy agents are cytotoxic. It means that they kill the cancer cells. To simplify it, think about standard conventional chemotherapy as high doses of treatment that's given to patients in one bolus dose. Then we wait for a few weeks and then give another high dose of treatment and then wait for the body to recover and so forth. While with targeted treatment, think about it as metronomic. It's continuous, small doses that accumulate in the body and work constantly on stopping the cancer cells from dividing and from growth. The first treatment that was targeted for lymphoma was developed in 1997, and it's called rituximab. Since then, and for the past 23 years, hundreds of approved targeted cancer treatment have been introduced to the field. There are different targeted drugs in lymphoma that work on different mechanisms of cancer cell survival, including antibody treatment, antibody conjugates, checkpoint inhibitors, CAR-T treatment, if you've heard of it recently, gained quite a bit of attention, cell signal blockers, proteasome inhibitors, immune modulators, epigenetic modifiers, and cell death inducers. I'll talk about some of them in detail next. The most common used treatment, targeted treatment in lymphoma, are monoclonal antibodies. They are laboratory produced proteins that have been engineered to specifically attack certain characteristic or antigens that are exhibited on the cancer cells. These can be very useful in treating cancer because they act specifically on that cancer cell. They can also enlist the help of our immune system to get rid of the cancer cells. And these drugs can be used in combination with other chemotherapy agents like R-CHOP in aggressive lymphoma. So the rituximab can be combined with CHOP, which is conventional chemotherapy agents. So how do these monoclonal antibodies work? This is the part of monoclonal antibodies. They can work by flagging the cancer cells. So they target, as we said, in rituximab, it's an anti-CD20 antibody. CD20 is expressed on the lymphoma, the B lymphoma cell. So they attach themselves to the CD20 antigen on the lymphoma cell. And by doing that, they coat the cell with the antibodies, and the immune system recognize these cells that are coated with the antibody and get rid of them and destroy them. They can trigger cell membrane destruction. So the cell membrane starts having holes in it, and then the cell get destroyed. They can directly attack the cancer cell and cause cell destruction. They can block the cell growth by blocking the signal that help that cell to survive and grow. And some of them can prevent blood vessels from forming. And if the cancer cell and the tumor in general doesn't get the nutrient and the oxygen that it needs to grow, it will shrink and die. So there are multiple ways that these antibodies can attack the cancer cells, and that's why they are highly effective. The monoclonal antibody rituximab is a cornerstone in the treatment of B-cell lymphomas. It works by bringing the effective natural killer cells to the B lymphoma cell and cause the cell to die. They can also enlist the help of the macrophages, which is a type of our white blood cells that can engulf the cancer cell and eliminate it. They can also cause a complex, as I described in my previous slide, on the surface of the B cell. This is the complex on the surface of the B cell, and that complex can be eliminated by our immune system, or they can cause holes by calling the complement system and the B cell, and the B cell dies. So multiple ways for that monoclonal antibody rituximab to effectively eliminate the B lymphoma cells. And indeed, in a landmark study that's been published some time ago now, we have shown that adding rituximab to the treatment in patients with aggressive lymphoma had improved the survival of these patients by 20%. And this is an extremely big step, or was an extremely big step in the treatment of lymphoma. Since then, rituximab has become a cornerstone, as I said, in the treatment of aggressive and low-grade B-cell lymphomas in combination with other chemotherapy agents. There are other monoclonal antibodies that target other antigens on the surface or other markers on the surface of the malignant B cells, like CD19, CD22, CD40, CD52, etc. And quite a few of these monoclonal antibodies are in development at the moment. One of these drugs is available in experimental level in many trials that are open here in New Zealand. They are called the B-Mind study. It's called tafasitamab. This drug, this monoclonal antibody, targets the CD19 that's expressed on the malignant B cells. And in a similar way to the activity of the anti-CD20 monoclonal antibody rituximab, it enlists the help of our immune system, effective immune system cells, to destroy the malignant or the cancer B cells. It also can bring, as I said, the macrophages, which can engulf and get rid, eliminate the malignant B cells, or it can directly lyse and destroy the B cell. Quite effective, similar to rituximab. Preliminary data in patients with relapse and refractory aggressive lymphoma showed that the overall response rate was up to 60% of the patients, with complete response in more than 1/3 of the patients, and a long duration of response of up to almost 3 years. These are extremely encouraging data, and we're waiting the final results of these studies. Another mode of action in targeted treatment is what we call an antibody drug conjugate. This is the mode of actions. We now know that antibodies are very effective in eliminating cancer cells, including lymphoma cells. Why don't make them even more effective? By attaching either a little radioactive molecule to the surface of these antibodies, or attaching the monoclonal antibody to a small, very toxic particle, or a drug like a chemotherapy, a drug that can deliver that drug straight into the cancer cells. And that's what has happened recently when these types of treatment have been developed. These are some of the antibody drug conjugate. One of them is called rituximab vedotin. It's an anti-CD30 monoclonal antibody conjugate that is attached to a very toxic molecule. It's called MMAE. And what happens is that these monoclonal antibody get internalized inside the cells that are CD30 positive cells. And once they are inside the cell, they start secreting those very toxic molecules, and the cells will die or burst. And it is a very effective way of targeting the cancer cells without causing much damage to the surrounding normal cells. Because CD30 is an antigen that's present in Hodgkin lymphoma cells, these are very effective in Hodgkin lymphoma, and this is a keynote study that led to the registration of the drug in the US. And they used it in patients, and you can see these patients who have relapsed or refractory Hodgkin lymphoma, and you can see most of the patient responded here. This is a waterfall graph. And recently, 2 years ago precisely, this is a very elegant study published from the USA. It's called the Echelon-2 trial. The drug was used in combination with CHOP chemotherapy in patients with CD30 positive T-cell lymphoma, and you see that the patient who received the brentuximab did slightly better than the patients who received the CHOP alone. So we are hoping to get these drugs in clinical practice soon. Another drug, monoclonal antibody conjugate, it's called protooncogenes vedotin. It targets the CD79B. It's also another marker on the tumor B cells. It gets internalized. It's attached again to the MMAE molecule, which is highly toxic to the cell, and the cell will burst and die. Studies that were done combining the drug with bendamustine and rituximab showed that 40% of patients with aggressive lymphoma have responded, and a median overall survival of 12.4 months versus 4 months in the patients who didn't get the polatuzumab. That led to its FDA approval in the relapse refractory lymphoma setting. Another way is targeting the immune system to help it wake up, because the cancer cells have evaded the immune system. So to help the immune system to wake up, recognize that the cancer cells are there, and attack them and eliminate them. There are few modalities of using the immune system as a targeted treatment against lymphoma. One of them is called checkpoint inhibitors, where the molecules, the treatment molecules block the cancer cell signal that stops the T cell from functioning, so the T cells start functioning again and attack the tumor cell. Another modality is to bring these T cells to the tumor cell to eliminate it using either bispecific antibodies or CAR-T treatment, which we've all heard about. The checkpoint inhibitors, as I said, it's a very effective modality. It's been found to be effective in certain types of cancer. What happens is that the cancer cell here secrete a molecule to its surface that numb our immune cell. So what these monoclonal antibodies or immune checkpoint inhibitors. They block these signals and free the T cells to circulate around the cancer cell and attack it. Some of these treatments, including nivolumab and pembrolizumab, have been found to be effective in subtypes of lymphomas, including Hodgkin lymphoma and the primary mediastinal B-cell lymphoma in the relapse setting. So we are hopeful we might be able to use them in the near future. Bispecific T-cell engagers or BiTE molecules have also a role in the treatment of lymphoma because they have two sides. So the antibody has two monoclonal, or that molecule has two monoclonal antibodies that joined together. One of them could be CD19 or CD20 that targets the B cells. The other one is CD3, which targets the T cell. What happened? These molecules bind to both the T cells and the B cells. So they bring our main weapon, which is the cytotoxic T cell, to the B cell and tell it, this is the abnormal cell. Can you help me eliminate it or destroy it? Mosunetuzumab is one of these particles. It has two sides. One of them is CD20. The other one is CD3. This is the CD20 molecule on the malignant B cell. And this is the mosunetuzumab. It brings the CD3 positive cell, which is our killer immune cell, at proximity to the malignant B cell and say, yep, this is the abnormal cell. Can you please help me eliminate it? Quite effective. We have a preliminary data from phase 1 studies that showed highly effective results, positive results in patients with both aggressive and low-grade lymphomas. And again, these graphs show you that the percentage of patients who have responded. 24 out of 29 indolent lymphoma patients and 17 out of 24 aggressive lymphoma patients achieved response and remained in remission up to 26 and 16 months after the initial treatment, respectively. CAR-T treatment, I believe that Dr. Rob Weinkoff gave a talk to LBC patients in a webinar a few weeks ago, and he detailed that therapy, and he has brought it to New Zealand. So I'm not going to spend a long time on it, but basically the patient's T cells are taken to a lab where they are educated and told to attack the lymphoma cells. They get reinfused again into the patient's body, and they start circling around and eliminating all the cancer cells. Highly effective treatment and very promising in aggressive relapse lymphoma patients. Another way of recognizing the cancer cell is through some of the pathways that take the signals, we talked about it, from the environment inside the cell and tell the cell to survive or die. So one of these signals is called BCR, or B cell receptor signaling pathway. This is required for the tumor survival and expansion. This pathway can be targeted by some molecules that stop that signal that lead to the cell death eventually. including PI3 kinases, Bruton kinase, and SYK inhibitors. The one that we have available here in New Zealand is the Bruton kinase inhibitor molecules or drugs, including ibrutinib, acalabrutinib, and zanubrutinib, in a clinical trial setting at the moment. There are a few trials that are enrolling patients to the Bruton kinase inhibitor drugs. They are especially effective in mantle cell lymphoma, Waldenstrom macroglobulinemia, and small lymphocytic lymphoma. Another group of molecules are BCL2 inhibitors. What is BCL2? It's a molecule that helps the cell to survive. It is a protooncogene. That molecule, when there is plenty of it and it is mutated, it stops the cell from dying. It stops the apoptosis because it's a surviving molecule or a cell survival molecule. What happens with venetoclax? It blocks the BCL2, the mutated gene that stops the cell from dying. Once you stop that molecule, then the cell will, death program will be activated and the cell will die. The preliminary data in lymphoma is disappointing, not as good as monoclonal antibodies, but ongoing studies are happening at the moment. The last slide is talking about epigenetic dysregulation in lymphoma. You might have heard this term, epigenetics. Epigenetic describes the changes of gene expression with no changes of DNA. It's the changes in the microenvironment of the cells and the enzymes that help the cell to survive. So if there are abnormalities or overexpression of some of these enzymes, then cancer develops and the cancer cell survives. Small molecules have been developed to inhibit some of these abnormal enzymes, and some of them have been approved for the use in lymphoma, like the histone deacetylase inhibitor. Histone deacetylase is one of these enzymes that I just talked about. So if you block it, then that leads to cancer cell death. One of these drugs is called romidepsin. It's used in T-cell lymphoma with variable success, with a response rate of 25 to 30%. I think the best way of using these drugs is possibly in combination with other targeted treatments. There are ongoing trials that are happening at the moment with this regard. So to summarize, there is a rapid improvement in our understanding of the molecular basis of various lymphomas, which have led to the development of many of the new treatments. Many of these new agents have shown promising results, and clinical trials are ongoing into clinical practice. And despite the early success as well as substantial and durable responses in some patients, there's still much work to be done, as you have seen. So many pathways for these cancer cells to survive and evade our immune system. And the more we study, the more we learn, the more effective we become in designing new treatment to target those specific genetic abnormalities within the cancer cells. And the greatest impact, as we've described, of these new treatment will likely come from the combination therapy, possibly in the frontline setting. But further studies are ongoing. There are clinical trials that are happening everywhere in the world, including in New Zealand, enrolling patients into these clinical trials using these targeted treatments. And we hope with more studies, more data will be generated, and we'll be able to determine the effectiveness as well as the best way of using these cancer treatment in the near future. Thank you.
Thank you, Dr. Issa. That was an amazing and informative talk. That was really a lot to think about. So thank you so much for sharing that with us. It's an invaluable resource going forward. We do have a couple of questions if you've got time to answer them.
I think we finished early because of the technical glitches that happened. I apologize. And I think we will have the link to these YouTube videos. Hopefully you can access them in your own time and look at these amazing images that have been put together on explain how these drugs work in real time. It was a real treat, but it didn't work. So apologies for that.
That's okay, it happens. I'm sure we can look into seeing if we can circulate some of those links as well when we send out the recording.
Thank you.
Yeah. So I just have one question here from an attendee. Do we need to have genetic testing to be able to determine what therapies are best suited for us?
Yeah, that's a really good question, because we are talking about the new era in cancer treatment and what we call individualizing cancer treatment. So we treat patients according to the cancer genetics, not the patient's genetic. We take the cancer cells and we look at the genetic make of these cancer cells. We already do some of these studies. We call them, if you've heard of them, karyotype, conventional cytogenetics, FISH studies, next-gen sequencing. So we sequence the cancer gene and we see what mutations these cancer cells have, and design the treatment according to that, what that particular individual cancer cell make is, rather than give R-CHOP to every patient, because some of these treatment target pathways that might be activated in one patient but not activated in another patient. We know nowadays that lymphoma is not the same disease in patients. They might have the same pathology, but they might have different genetics. So definitely the future will be designed treatment, targeted treatment according to the genetic mutations of each individual lymphoma, and not one size fit all.
Thank you, that's good. Just had another question here. If I think one of these therapies would be good for me, can my hematology team find special funding?
Some of these drugs are available on compassionate funding use by the drug company. So your hematologist can contact the drug company and see whether they are available in New Zealand on compassionate use. Some other targeted therapies are available in the trial setting, so you can be enrolled into a clinical trial and get the drug through that pathway. But quite a few of them are still experimental. The final results are not back. So depending on what type of lymphoma you have and what stage of the disease treatment you are at the moment, it might be suitable or maybe not suitable to use these treatment. The fact that they are available doesn't mean that we have to use them. They are used in suiting settings. So This discussion is best initiated with your hematology team and with your treating consultant, and see whether your lymphoma is suitable for certain type of treatment, whether you need it or not.
That's great, thank you. And I actually had a quick question. With all of these new advances and all of these, I guess, targeted therapies, where do you see the future of traditional chemotherapy? Do you think that's something, you know, in 10 or 20 years we might not be using as much, or do you think that will always be part of the treatment plan?
In 10 years, I think it might still be part of the treatment plan, because it targets rapidly dividing cells. And as I said, these targeted treatment are cytostatic, not cytotoxic. So you want to cytoreduce, you want to reduce the number of the tumor cells, the bulk of the tumor, so to speak. So chemotherapy, radiotherapy will still be available in 10 years, but I see that these treatments get combined with chemotherapy, and maybe also used in the maintenance setting. Once you debulk the tumor, got rid of the main lymph nodes or mass, then these treatment can be used on continuous in a continuous manner, just to prevent the tumor from growing back and help prolong the remission status of the patients. Maybe in a 20 years time, Melody, we can do the personalized treatment, i.e. we take the tumor sample, we do full genome sequencing, see what abnormalities that particular lymphoma has, what gene mutations it has, and then combine different. I don't see a one single agent being used. You combine different agents. The reason why I'm saying that is because to develop cancer, you need multiple mutations on multiple levels. So it's not only the BCR pathway that's mutated, or the BCL2 gene, or the BTK. There are multiple pathways that contribute to the development of cancer. So you need multiple targeted drugs on multiple levels to block the cancer cell from surviving. And so possibly in few decades from now, when we have more molecules to target, more pathways, you can look at the gene mutations and you say 1, 2, 3, 4, 5 genes are mutated in this patient. So you give 1, 2, 3, 4, 5 targeted therapies to target these lymphoma cells. I don't see it happen in the near future, but I certainly see the targeted treatment being combined with conventional treatment, possibly more than one targeted agent, and then using them in the maintenance setting.
That makes sense. Thank you for that. That's all the questions we have for now, unless there's any kind of last minute ones floating out there. Otherwise, I think we might wrap up there. Thank you again, Dr. Issa. That was really wonderful talk, and I look forward to going back and kind of rewatching and letting it all sink in a bit more as well. Thank you everyone who has joined us this afternoon. And as I said, it's all been recorded. It's all going to be uploaded onto our YouTube channel for your continued viewing pleasure. So thank you everybody, and have a great afternoon.
Thank you, Melody, and thank you everyone for logging in.